everybody, it's Mr. Miller again. So this lesson today is all about active and passive transport. In the previous lesson, we talked about how cells need stuff, and there's stuff inside the cell and there's stuff outside the cell. And some stuff needs to leave the cell, and other stuff needs to come in. And we talked about how the cell membrane, which is the part that goes around the cell, regulates what comes in and what goes out. And we said that in the previous lesson, we said that your body wants to move things from where there's a lot of that stuff to where there's a low amount of that stuff until there's about the same amount of stuff inside the cell as there is outside the cell. And this direction that your body wants to move things in so that it can balance out how much is inside versus outside, in this case, out to in, because it always flows high to low. This is what we call the concentration gradient. Now, we mentioned that if you want to move something in the same direction as the concentration gradient, it's just like if you wanted to move in the same direction as a river. All you have to do is hop in and do nothing and let the river carry you. It requires no energy, and that is called passive transport. When you go with the gradient and there's no energy involved. However, if you want to do the opposite and you want to move something in the opposite direction of where it wants to go, high to low, if you want to move it from low to high, you have to go against the flow of the river, which requires energy and effort. And that's called active transport. That goes against the flow or the concentration gradient, and that does require energy. All right, now that was just a quick recap of what we talked about last time. Now we're going to get into what we're talking about this time, which are the different kinds of active transport and passive transport. Let me explain the difference between the different kinds. So once again, we have our cell. That's a pretty good circle. I think I might have just drawn a perfect circle. And I have video evidence, which is even better. Anyway, I can't get over how good that circle is. Okay, so active and passive are the two types, but there's different ways that we can move things in and out. Let's first talk about active. So we know that active, let's say that we have a high amount inside and a low amount outside of stuff, whatever the stuff is, okay? Doesn't matter what the stuff is. The concentration gradient is going to go from high to low, or in other words, from in to out. Okay? So if we're wanting to move something into the cell against the concentration gradient, it's going to require energy. And there's a couple of different ways that we can do this, and it really just depends on the size of the thing that we are trying to move, okay? Now, as we have learned, embedded into the cell membrane are these what are called transport proteins. So these transport proteins, another name for them is pump. So once again, the concentration gradient is going from in to out. But let's say we want to push something against that gradient and go out to in. Well, that pump would be the one to do it, but it requires energy. And that energy comes in the form of what we call ATP. And you always see ATP drawn like with a bang symbol around it. ATP is just energy, okay? So you have to put energy into this process, just like you have to put energy into pumping a bike tire full, because you're going to pump things against where it wants to go. In other words, swim against the current of the river. So that's one way to do it, is through a transport protein or a pump. But let's say that whatever it is you're trying to bring into the cell, there are some things that are too big to fit through this transport protein. It's just like a door on the wall. You can't come through the wall, you have to come through the door. But let's say you're too big for the door. Well, then you got to break the wall down and build it back up. So let's say this is the thing that we need to bring into the cell. It's really big, right? It won't fit through there. So the only way to do it is to literally break down the wall, get the thing in, and then build the wall back. Now the cell membrane is very squishy and fluid. So this isn't actually a big deal like you might think it would be. So step one kind of looks like this. It's outside the cell, and then step two, the thing goes and it makes contact with the membrane, and the membrane sinks in and wraps around the thing that you're trying to bring in. And then it will wrap all the way around and pinch and bring this in to here. And then this will pop back into place. There. And that process of bringing in things that are too big for the cell, for the transport proteins, is what we call endocytosis. It still requires energy, so it is a form of active transport. Okay? But the difference between this and moving it through a pump is just that it's too big to go through the pump. That's the only difference. That's why I said size. The difference is just the size of the thing you're trying to move. Okay, so that's endocytosis, bringing it in. But if you need to move it out, the same thing happens, just in reverse order. Okay, it moves out, and then this keeps going. The cell membrane pops right back into place, right behind it. And that is called exocytosis. Endos bringing it in, exos making it exit. So, endocytosis and exocytosis are the two other kinds of active transport that require energy that I wanted to show you, and it's because they're too big to go through the transport protein. All right, so that was active transport. So now let's talk about passive transport. Let's see if I can make a perfect circle again. Woo, that's not as good as the first one. All right, but passive transport is, let's say you have high in and low out. Well, we know the concentration gradient's gonna go high to low, so that's the direction of the current. And passive transport is gonna move stuff in that same direction. Now, once again, there's three different kinds that I'm gonna tell you about. There's diffusion, then there's facilitated diffusion. Make that 1 and 2 so you don't get confused. Did I say three different kinds? Well, I meant two different kinds, sorry. So we go from in to out, high to low. Now, once again, the difference between diffusion and facilitated diffusion is how big the thing is. So let's say that the thing is teeny tiny. Well, this cell membrane is actually made up of these guys called phospholipids. If you were to zoom in on the cell membrane, that's what you would see. It's actually 2 layers of phospholipids. But these guys are fluid. They can be moved and pushed around. So if something is tiny enough and it needs to go from inside the cell to outside the cell, or vice versa, if it's small enough, like oxygen or carbon dioxide, these are tiny molecules. They can actually just squeeze right through there and go in any direction, in or out, that they need to at any time that they need to, because they're that small. And that's what we call diffusion. So diffusion is just tiny particles that can fit through or squeeze between the individual lipids that make up the cell membrane. But let's say that the thing is too big. Now, if we're zoomed in, this is too big, it's not going to work. But if we're on the zoomed out version, it may only be this big, right? Now, in that case, it's too big to leave through the cell, so it's just going to bounce around in here, right? However, we can use the same transport protein to go from inside the cell to outside the cell, or in other words, with the concentration gradient, which is passive transport. But because we're too big to squeeze through the lipids in the membrane, we have to go through the door, which is the transport protein. And it's still diffusion, but because it's diffusion that needs some assistance from the transport protein, we call it facilitated diffusion. It's still diffusion, it's just diffusion through a transport protein. Why is it going through the transport protein? Because it's too big to just squeeze between the individual lipids that make up the cell membrane. Okay, and the last thing I need to tell you is, real quick, you're going to hear the word osmosis. All osmosis is, it's the same exact thing as diffusion, only it's diffusion with water. If it's anything else that's doing the diffusing, it's just called diffusion. If it's water that's doing the diffusion, it's called osmosis. Why does it have a different name? Why don't we just call it diffusion with water? I don't know. That's a good question, and I don't have a good answer. But all I know is that when it's water, it's called osmosis. And so if you see the word osmosis, just know they're just talking about diffusion, not facilitated, regular diffusion. Because water is one of those tiny particles that's tiny enough, or tiny molecules that's tiny enough to squeeze through the individual lipids in the membrane. Okay, and the last thing is why. why do cells move things in and out? Well, they move things in and out because they want to maintain what we call homeostasis, which is just a stable internal environment. That's all homeostasis is. And the way it does that is by trying to obtain what we call equilibrium. Okay? Equilibrium is when you have the same amount of stuff outside as stuff inside. Not necessarily the same amount, it's the same concentration, but we'll talk about that later, okay? I don't want to put too much on you right now. But for, let's just say stuff for right now, amount of stuff. Okay, so when the cell is happy, it's called being in homeostasis. It has a stable environment. And when it has the same amount of stuff outside the cell as inside the cell, that's called being in equilibrium. The two aren't always the same, but just know the difference between the two. And the cell has stuff, there's stuff outside the cell, there's stuff inside the cell. Some stuff in needs to go out, some stuff out needs to come in. And the reason it does it is because it's trying to obtain a happy environment, homeostasis. And that is it in a nutshell for this video on active and passive transport.